soul back. So
It's
that's the
bad.
only
All
one
right,
that's
guys, we're
bad.
gonna get started. We got a lot to cover on your little time. I said no phones. It's in the syllabus.
The ear is comprised of three parts: the outer, middle, and inner ear. The outer ear collects sound waves in channels. It consists of the auricle, the external auditory canal, and the eardrum. The auricle is a flap of cartilage covered by skin. The external auditory canal is about 2.5 centimeters long, and at the end of it is the eardrum, which is also known as the tympanic membrane. The middle ear conveys sound vibrations to the oval window. It consists of the malleus, incus, and stapes. The malleus, incus, and stapes allow the sound waves to travel through the ear, and at the end of the stapes is the oval window. Inner ear is divided into the outer bony and inner membranous labyrinth. The bony labyrinth consists of the semicircular canals, vestibule, and cochlea. The membranous labyrinth is surrounded by perilymph, which is a series of epithelial sacs that contain endolymph. The cochlea consists of the cochlear duct, the scala vestibuli, and scala tympani. And the vestibule consists of hair cells that contain sensory neurons for hearing.
The process of hearing begins with the auricle directing sound waves into the auditory canal. The sound waves hit the eardrum and causes it to vibrate because of the intensity and the frequency of the waves. The vibration of the eardrum travels to the malleus, incus, and the stapes. As the stapes vibrates, the membrane of the oval window is pushed. The oval window vibrates more than the eardrum and causes fluid pressure waves in the perilymph of the cochlea. The oval window pushes on the scala vestibuli, and the round window bulges toward the middle ear. The pressure waves deform the walls and push the vestibular membrane back and forth, creating pressure waves in the endolymph inside the cochlear duct. The pressure waves in the endolymph cause the basilar membrane to vibrate and move hair cells. The bending of the hair cells produces receptor potentials and nerve impulses.
Nerve impulses travel through the axon of the sensory neuron, known as the cochlear branch of the vestibulocochlear nerve. These axons synapse with the cochlear nuclei in the medulla oblongata. At this point, axons can continue to send nerve impulses multiple ways. Some axons from the cochlear nuclei cross over the medulla, flow up the lateral meniscus, and terminate in the inferior colliculi in the midbrain, and in the superior olivary nucleus in the pons. Axons from the superior olivary nuclei also travel up the lateral meniscus tract and end in the inferior colliculi. Nerve impulses are conveyed from each inferior colliculi to the medial geniculate nucleus. These impulses end at the primary auditory areas of the cerebral cortex in the temporal lobe of the cerebrum.
Hair cells are specialized sensoreceptor cells located in the inner ear. They are cylindrical or flat-shaped and are equipped at the apical end with a bundle of sensoreceptors known as stereocilia. An orchestra of chemical synapses occurs in order to get nerve impulses from the ear to the brain. The neurotransmitter glutamate is released into the synaptic cleft, which in turn causes nerve impulses in the sensory neurons.
At the chemical synapse, the electrical signal is converted into a neurotransmitter that acts on the target cell. Because it's a chemical synapse, it will be released from the vesicles and binds to a receptor. Compared to electrical synapse, which occurs at a gap junction. When these cells are released, they act as paracrine signals, in which they act on cells near or around the area. So with paracrine signaling, it will attach to the receptor and your desired result would happen.
Action potential is a brief, large depolarization, and it travels long distances. However, it does not lose its strength along these long distances. Action potentials occur on the trigger zone of an axon. This trigger zone is right around the same area as the axon HELOC. Action potentials are initiated by a graded potential occurring above the threshold around the trigger zone. This stimulus would open the voltage-gated ion channels of sodium, and it would cause depolarization of the cell, and it would start the action potential. It allows the sodium to diffuse within the axon and to depolarize it to about positive 30 millivolts. and that's where your action potential would occur. This voltage-gated sodium channels would then close, and then the voltage-gated potassium channels would then open. And with the opening of these channels, potassium would diffuse out of the cell, and that would cause the repolarization of the cell. This repolarization causes it to a negative voltage value. The potassium would then continue to go and to flow out of the cell, and this would cause the refractory period, or more known as hyperpolarization, because it is a extremely lower negative value of the cell being caused.
The membrane potential of a resting cell is around -70 millivolts. This number indicates the electrical potential difference between the outside and inside of the cell. A negative number, like the one of our cells, tells us that the inside of the cell is more negative than its extracellular surroundings. This state can be achieved by the potassium-sodium pump, which pumps more sodium out of the cell than potassium in. Cell membranes are more permeable to potassium and readily have open leak channels for this ion. Other ions can be controlled by voltage, such as calcium, or by chemicals of ligand-gated channels.
So class, as a matter of fact, since you want to be on your phone, tell me how sound travels through the ear.
Well,